Let's move to the topic which involves few optical instruments. Although this topic is not part of IIT JEE syllabus, but it forms the part of syllabus of AIEEE and various entrance examination, that is the study of microscope and telescope. The questions which have been asked, they are very simple and straightforward, and they are just based on the basic understanding of these two instruments. And it will be better for IIT JEE also, because there are some problems which involve a mention of these two instruments. So we'll be just going through the basic points of these two optical instruments, that is microscope and telescope. To start with, let's take up first simple microscope. Now the principle of a simple microscope, which is also known as a magnifying glass, is very simple, and it is based on the point that when an object is placed between a convex lens and its principal focus, an erect, virtual and magnified image is formed on the same side as the object. A single convex lens of short focal length thus used constitutes a simple microscope, which is commonly known as magnifying glass or reading glass. Now let's understand the formation of image by simple microscope. The figure on the screen shows a small extended object AB, which when viewed by an unaided eye cannot be seen distinctly. A convex lens is then placed between the eye and the object, so that the distance u of the object from the lens is less than the focal length f of the lens. A virtual, erect and magnified image A', B' will be produced. By adjusting u, the image is set at least a distance of distinct vision, that is capital D, and its value is 25 centimeter from the eye, so that the image becomes most distinct. The rays of light forming the virtual and magnified image A', B' of the small object AB, as seen through lens by placing the eye very close to it, is shown in the figure. Now there is one term associated with microscope, and it is magnifying power or magnification. The magnifying power of simple microscope is defined as the ratio between the angle subtended by the image at the eye to the angle subtended by the object at the eye, when both image and the object are situated at the distance of distinct vision. Let's make calculation, and using the figure shown on the screen, if theta I is the angle which the image A', B' subtends at the eye, and theta naught in subscript, and theta naught is the angle which the object AB, which is equal to CB', subtends at the eye, when both are placed at the distance, that is at the least distance of distinct vision D, then we can write magnifying power as theta I over theta O, which we can write from small angle approximation as tan theta I over tan theta O. Using the basic definition of trigonometric ratio tan theta, we can write magnifying power capital M as AB over OB divided by CB' over OB', and this on rearrangement gives us OB' over OB, and here we have to make use of the fact that AB is equal to CB', and that is equal to minus D over minus U, and that gives us finally capital D over U. Since the virtual image A'(B' is formed at the least distance of distinct vision from the lens, therefore V is equal to minus D. Now using the lens formula, we get the equation, that is 1 over minus D plus 1 over U is equal to 1 by F, and this on rearrangement gives us the value of D by U, which is 1 plus D by F. Hence the expression for magnifying power of simple microscope is 1 plus capital D by F. Therefore a convex lens of shorter focal length yields higher magnification. Now we can have two specific cases. When the final image is formed at infinity, then magnification is given by M infinity in subscript is equal to D by F. Second, if the lens is kept at a distance x from the eye, then M D in subscript, that is magnification for image formed at least distance of distinct vision, will be equal to 1 plus capital D minus X divided by F. Similarly, the magnification for image formed at infinity will be D minus X divided by F. Now this simple microscope or magnifying glass is used by astrologers to read fate lines of hand, biology students to see the slide, watch repairers to locate defects, and defective department to match fingerprints. Now we'll take up a compound microscope, which is a modification of a simple microscope. The magnification produced by a simple microscope is generally not sufficient to give a detailed view of two very closely situated objects. A combination of two convex lenses is employed to have a larger magnification. The instrument which is designed on this fact is known as a compound